quick announcement. Thursday at noon in CST 103 will be the first biology colloquium of the semester, and Dan Atwater will be talking about his research regarding the dangerous world of plant ecology. So you're all invited to this biology colloquium. Okay. That's the announcement. The other announcement is that I have your problem sets graded, so at the end of class today, I'll hand back your problem set. The grades have been posted to Moodle as well. So, previously we talked about the heart anatomy. We used some of the names. Rather than go through it again, what I'm going to do is link to this YouTube video, which you can, cut the URL out and paste into your browser. But that's what I'm going to do now. This animation will walk us through quite quickly the flow of blood in the heart using some, not all, of the anatomical terms that we talked about last time and that you should be studying from the coloring book for the quiz due on Sunday. So there's a quiz on Sunday. I guess this is another announcement. Quiz due on Sunday. Closed book, so please resist the temptation and make sure you can resist the temptation by just putting everything away. Leave it at home when you come into CST to pick up the quiz. You'll come into CST, you'll pick up the quiz, you'll just sit down somewhere in CST and take it, and then turn it back in. It shouldn't leave the building. The quiz will cover both anatomy and physiology. It's about half anatomy, half physiology. Okay, so right now what I want to do is use this video, which we can play more than once, because it does move pretty quickly, through the flow of blood in a heartbeat.
The normal heart has two sides, a right side and a left side, and four chambers, the top receiving chambers, or atrium, and the lower chambers, which are thick-walled pumping chambers, called ventricles. Red blood cell will come from either the superior vena cava or the inferior vena cava and enter into the right atrium. The blood then flows across the tricuspid valve to the right ventricle. The right ventricle then squeezes and ejects that blood cell into a vessel called the pulmonary artery. The pulmonary artery splits into two vessels, each going to the lungs. As that red blood cell makes its way through the lung, it returns through the pulmonary veins to the left atrium. That blood is now oxygenated. It's picked up oxygen, then goes across the mitral valve into the left ventricle, which does most of the work in terms of delivery of blood flow to the body. That blood cell is now ejected into the aorta to some organ, or muscle, or skin in the human body.
Okay, so that is a 1 minute.
The normal heart.
A 1 minute review. Eventually it'll be a review of the flow of blood in the heart. So let's do a little stop action, and let's see where our narrator is. Two sides, a right side and a left side. So right. Remember that when you're looking at something anatomically, you're always looking at somebody else's heart. So this is this person's right side, and this is this person's left side. Even though it's your right, it's this heart's left. Don't get switched up. Okay, so right and left side.
Has two sides, a right side and a left side, and four chambers. The top receive
Atria, right atrium, left atrium.
chambers, or atrium, and the lower chambers, which are thick-walled pumping chambers, called ventricles.
Ventricles. Right ventricle, left ventricle. This cartoon is not accurate. It shows the wall of the right ventricle being approximately the same thickness as the wall of the left ventricle, and you saw in the pig heart and in the human heart, that's not even close to true. So the right ventricle has a much thinner wall than the left ventricle, and both ventricles have much thicker walls than both atria.
Red blood cell will come from either the superior vena cava or the inferior vena cava.
Superior vena cava, inferior vena cava, drain into the right atrium.
And enter into the right atrium. The blood then flows across the tricuspid valve.
The tricuspid valve is open during the period of time when the ventricles are not beating. We'll talk about this. So in between ventricular contractions, the tricuspid valve is open and blood is flowing. Blood flows from the atria into the ventricles. This is refilling the right ventricle. Through the tricuspid valve, you should know the name of the valve, know the name of these strings, chordae tendineae.
To the right ventricle. Right ventricle then.
Know the name of these nipple-like muscles that connect chordae tendineae to the that connect muscle to the chordae tendineae. So those are papillary muscle. And it also illustrates a structure here, this sort of meshwork of muscle called the trabeculae carneae. Those are all on your list. So use your list and the images in the coloring book and the images in a video to process and learn the anatomical details.
Squeezes and ejects that blood cell into a vessel called the pulmonary artery.
Okay, so when the right ventricle contracts, it squeezes blood out of it. So that squeezing closes the tricuspid valve so that the blood does not flow backwards, and opens what's the name of this valve here? Pulmonary semilunar valve. So that pressure, it's the pressure in the right ventricle that closes the tricuspid valve and opens and forces blood out the pulmonary semilunar valve into the pulmonary trunk and then into the pulmonary arteries.
Pulmonary artery splits into two vessels, each
Pulmonary
going
arteries. Okay.
into the lungs. As that red blood cell makes its way through the lung, it returns through the pulmonary veins.
Pulmonary veins return blood from the lungs into the left atrium,
To the left atrium. That blood is now oxygenated. It's picked up oxygen, then goes across the mitral valve.
When the left ventricle is relaxed and refilling, blood flows from the left atrium through the bicuspid or mitral valve into the left ventricle. And then
Into the left ventricle, which does most of the work in terms of
work. Then it pumps, it contracts, it does work. It contracts, that contraction of the left ventricle closes the bicuspid or mitral valve to prevent regurgitation of the blood from the ventricle back into the atria. We don't want that. And that pressure opens, what's the name of this valve? The aortic semilunar valve. Opens the aortic semilunar valve and forces blood out into the aorta.
delivery of blood flow to the body. That blood cell is now ejected into the aorta to some organ or muscle or skin in the human body.
Okay, so go through, you can probably you can slow this down and run it. Let's just see, can we run it at, sure, we could run it at twice normal, no, that wouldn't be helpful. Run it at quarter speed. Let's just see what happens, how weird this is if we run it at quarter speed.
No.
You'd have to narrate it yourself, but that might be the best thing to do, is to turn off the sound and run it slow and narrate it yourself with the things that are happening and with the anatomy. That would be a reasonable thing to do. So here's an image of the heart, still image, that you can use to learn the names, because this is labeled more clearly than it's labeled, the video clearly isn't. This shows slightly better that the left ventricle has much more muscle than the right ventricle, but it doesn't show it very well. But you've seen the real thing. You know what it looks like. You know that you can, these tubes here, arteries to head and arms, you can annotate with the actual names. Brachiocephalic, left common carotid, left subclavian. Those level of details could be on the exam because I've used it several times in class. It is in the coloring book assignment, even though it's not one of the things you color, it is part of the labeling of the figure, and it is on your list. So those are fair game. Not everything on your list will be on this quiz. So for example, we haven't got into the arteries of the digestive tract. So if I haven't said it in this room, and it wasn't on the coloring book, it's not going to be on the quiz. It will be on the exam at the end of this section. I'm happy to answer questions as you're preparing for the quiz that are, do we need to know, da-da-da, and I will tell you whether that's a good idea to know or not. So now we're going to work on moving from anatomy to physiology, the mechanism of the heartbeat. That's what we're trying to do now. Using the anatomy to help us have a more detailed discussion of the mechanism of the heart. So the first thing to remember is that the heart has an intrinsic pacemaking activity. Its activity, its rate, and its strength is modulated by the nervous system. You know that the sympathetic nervous system, when you're stressed, makes your heart beat faster and stronger. You may not know, but now you know that the parasympathetic nervous system, which is the rest and digest nervous system, so there's the fight and flight nervous system, which is sympathetic, and the parasympathetic is rest and digest. That has mechanisms for slowing and decreasing the strength of the heartbeat. But the heart beats intrinsically. It doesn't need a nervous system to beat. And you probably could have figured this out if you think about a transplant. We transplant hearts from one person to another, and the heart beats. But we're not connecting the nerves. We're only connecting the plumbing of that transplanted heart. So we're going to talk about the intrinsic mechanism, intrinsic, that is to say, within the heart mechanism for making a heartbeat. It's an electrical system, right? Because the heart is a muscle, and we need action potentials to trigger contraction of the heart muscle. And action potentials are an electrical phenomenon, so we need to talk about electrophysiology. That's what we're doing right now. So the heartbeat starts in the sinoatrial node. This is a part, a little region of the right atrium, the superior right atrium, and you can't see it. It's too small, it's buried within the wall of the right atrium. It's an anatomical name, but you can't see it on an anatomical diagram. The sinoatrial node is where the heartbeat starts. So okay, let me just do that and talk about, well, let me go to the next slide and see how this works. So the heartbeat starts in the sinoatrial node. Now, what does that mean? It means the sinoatrial node fires action potentials, fires an action potential. One action potential is going to get the heartbeat started. Not a whole series of them. A whole series of action potentials would be a whole series of heartbeats. One action potential is going to trigger one heartbeat. One action potential then spreads, that's what this yellow cloud and these yellow arrows in this cartoon from your textbook is illustrating. That action potential spreads throughout atrial muscle. Why does it spread? Because remember, all the muscles in cardiac muscle are connected by gap junctions. So the action potential in the SA node spreads throughout the atrium, both the right atrium and the left atrium. What happens then? What does an action potential in atrial muscle cause? Trigger?
Opening up the valves.
It will, kind of, need something between the electrical event of the action potentials and the valve changes. There's something in between. Michael? Oh, Kendra? Whoever? Somebody?
Is calcium released?
There is calcium. Good. So there's going to be calcium influx through voltage-gated calcium channels, calcium release from the SR, and then contraction. So this yellow wave here is supposed to represent atrial muscle that is contracting. And that is going to force blood to flow from atria through the open tricuspid valve and bicuspid valve into the right and the left ventricle. I'm going to use a word now that I haven't used yet, but I've been resisting, so I'm going to stop resisting using that word. The word is systole. Systole is the word for contraction, for pumping. And so what we just described was atrial systole, because the atria just contracted. Have we described yet ventricular systole? No, we haven't gotten there yet, so that's where we're going. If you ever go to a lecture and somebody calls it systole, you know they don't know what they're talking about. Because that's not the way you pronounce systole. So after the electrical activity spreads throughout the atria, it spreads into another structure here. This little yellow bulb here is supposed to represent something called the atrioventricular node. So SA stands for sinoatrial, and AV stands for atrioventricular. It's another very small, can't see it on the heart unless we do microscopic anatomy. And it's now it's in the right atrium, but towards the base of the right atrium. So there is this thing called the AV node, and I'll explain later what that does. What it does is it enforces a delay between atrial systole and ventricular systole. The heart is designed to pump the blood from the atria into the ventricles, and then pump the blood from the ventricles to either the lungs on the right side or the body on the left side. And the AV node is what does that delay between atrial systole and ventricular systole. So now we have to describe ventricular systole. Ventricular systole is happening while the atria are relaxing, but let's not worry about that. So ventricular systole, the electrical activity spreads from the AV node into the interventricular septum, into the ventricular apex. I'm using anatomical words that are good to know, and then into the ventricular walls. So it goes from septum to apex to walls. But eventually, all of the ventricle has fired an action potential. Calcium has flowed in, calcium is released from the SR, they have contracted, and we have ventricular systole. In lab this week, we're going to be turning those things into the electrocardiogram, but let's not worry about the electrocardiogram until Thursday. Let's think about the electrical events in the heart itself. Then, after the ventricles contract and pump blood, on the right side they pump the blood through the pulmonary semilunar valve into the pulmonary trunk, and on the left side it pumps blood through the aortic semilunar valve into the aorta. And that video is walking through it sort of sequentially, because it's talking about one red blood cell that moves, through the right side and then through the lungs and then through the left side. But right ventricular systole and left ventricular systole are happening at the same time. Then after systole, we have to have relaxation. Relaxation, why? In order for the heart to refill. We have to refill it for every beat. And the interval between heartbeats, the relaxation refilling period, is called diastole. So diastole is resting, refilling, then atrial systole, then ventricular systole, then diastole again, over and over again, about once every second. Yes?
So we don't differentiate between atrial and ventricular diastole, it's just diastole for the whole heart?
Pretty much. Other questions? So again, the reason why these, I want to just make it clear, why is electrical activity spreading from SA node into atria? Why is it spreading, I need to use some more anatomical terms. Why is it spreading from the AV node into these two branches called bundle branches, left and right bundle branches? It's spreading into the septum, it's spreading into the ventricular apex and into the wall. Why is it doing that? It's because all the cells are connected to their neighbors by gap junctions. And so an action potential in one cell spreads to the next cell. And so contraction in one cell triggers contraction in the next cell. So I'm going to do one thing here. I would like to show you another. Bless you. I would like to show you another animation. This is an animation that was designed to teach medical students about the electrocardiogram. We're not going to get to the electrocardiogram today, but the animations are helpful. So let's just look at this. And it repeats so I can talk about it. In this cartoon, the blue structure is the SA node, sinoatrial node, and it fires an action potential which spreads throughout the atria, the right and then the left atria, triggering that action potential in the atrial muscle, voltage-gated calcium channels, calcium release from the SR, calcium binding to troponin, all the stuff, good stuff you know, triggering contraction of the atria. So this you can see the wave of electrical activity spreading over the atria, triggering atrial systole. You can also see the AV node, the left and the right bundle branches, and these purple wormy things are Purkinje fibers. So, let me write that one out, Purkinje. I'm sure that's how we say it in the United States, Purkinje fibers. Purkinje was a Czech anatomist, and I'm sure in the Czech Republic this is pronounced differently, but we call it Purkinje. So, you can see that action potentials, when they reach the AV node and are delayed there, are eventually going to spread through the AV node, the bundle branches, and the Purkinje fibers, and then into ventricular muscle. Okay, so let's go back to the main menu. Okay, so after the electrical activity spreads throughout the atria, you might think, okay, it's going to spread throughout the ventricles. It's going to spread from here, it's going to spread through here, it's going to spread all through here. You might be wondering, why does it only go through the AV node? And the answer to that question is that the atria are electrically isolated from the ventricles. There's a layer of fat and connective tissue that prevents the spread of electrical activity from atria to ventricles, except at the AV node. The AV node is the window from the atria to the ventricles as far as electricity is concerned. There's, except for the AV node, the atria are insulated, isolated from the ventricles. So, electrical activity spreads throughout the atria. It does not spread from the atria to the ventricles here. It does not spread from the atria to the ventricles here. It only spreads from the atria to the ventricles through the AV node, the bundle, this has got a name which I'll mention later, bundle branches, see how it's spreading down the bundle branches there into the interventricular septum. Skip a bunch of things because it's more detail than we need. It's the same thing. Okay, so now, after it has spread through the AV node, the bundle branches, you can watch this wave of activity. Spreads in the interventricular septum, then the septum, then the apex, then the right and the left wall. That's the wave of electrical activity that triggers ventricular systole. Why do you need to know that level of detail? Because it's that level of detail which will allow you to interpret the electrocardiogram. And it's the electrocardiogram that provides information about that level of detail. And the electrocardiogram is a clinical tool that some of you will use, and the rest of you might want to know just because you're getting an ECG being recorded on your body. And that's why we're doing it at this level of detail. Because you want to understand, to be honest, I want you to understand the electrocardiogram. Okay, so now having talked about the spread of action potentials, I want us to get into the details, the channel details that make the action potentials that I've been talking about. Okay, so now we're getting into the channels that create the action potentials that create systole, whether it's atrial or ventricular. So let's talk about the SA node as a pacemaker. Now I need to build on the knowledge that you have from self-phys about action potentials. This action potential in the AV node is different from the action potentials that you're going to see in the rest of the heart and in from the nerve that you learned about in self-phys. So the first thing to realize is the SA node is the pacemaker. So it's firing an action potential on its own. It doesn't need any stimulus to fire an action potential. It is an intrinsically active pacemaker activity, intrinsically active pacemaker that fires action potentials spontaneously, about once a second. Faster if you're exercising, slower if you're sleeping, but let's just say about once per second, 60 beats per minute is a normal heart rate. That's a reasonable guess for a heart rate. My heart is probably beating closer to 75, yours might be beating closer to 55. Around 60 beats per minute, 1 beat per second. Intrinsic. So how does it make its own action potential spontaneously, intrinsically? So there is this in self-phys and in AMP1, we talked about a resting membrane potential. Pacemaker cells don't ever sit at the resting membrane potential. They go negative, just like you would at a resting membrane potential, but then they spontaneously depolarize in this sort of ultra slow, it's very slow, ultra slow depolarization. So the membrane potential repolarizes from the previous beat, because there's a beat back there and there's a beat over there. It's happening every second. It repolarizes, and then it's ultra slow, super slowly, I mean by ultra slow, I mean this is about a half a second. It takes about a 3/4 of a second for it to depolarize. And it depolarizes spontaneously to threshold. And then when it reaches threshold, it fires an action potential. And that's the action potential that spreads into the atrium. The channels that are responsible for the repolarization, just like in heart, just like in muscle, and just like in nerve, are voltage-gated potassium channels. Voltage-gated potassium channels, open, potassium flows out, leaving the inside negative. Now what happens? There is a type of channel called IH. Your book calls it IF. It's the same thing. IH, the H stands for hyperpolarization. It's a channel that is sodium permeable that is activated by hyperpolarization. So when the membrane polarizes, because of voltage-gated potassium channels, IH channels open, sodium flows which way? In, and that sodium influx causes depolarization. That depolarization then triggers the activation of your normal voltage-gated calcium channels, the ones that we've been talking about so previously. Which way does calcium flow? Calcium flows in. That calcium influx causes further depolarization. So that's the mechanism of, well, one more thing. The potassium channel, so I need to jump to the other end. Remember that potassium channels were responsible for the repolarization, but they are opened by depolarization and closed by hyperpolarization. So they're open here, they're causing the repolarization, and then the potassium channels close. So the closing of potassium channels the opening of a sodium permeable hyperpolarization activated channel and the opening of a voltage depolarization activated calcium channel cause this ultra slow, also known as pacemaking potential. That's the mechanism, the molecular mechanism for pacemaking. It spontaneously reaches threshold, and it spontaneously fires an action potential. But here's a twist. Unlike the action potentials in nerve, and unlike the action potentials in skeletal muscle, the action potentials in nodal pacemaker cells, AV node, don't use voltage-gated sodium channels for the upstroke of the action potential. They use voltage-gated calcium channels. So, and these are the same channels. So why is this slow and this fast? We've reached threshold. This is a few calcium channels open, and this is a lot of calcium channels open. Calcium is flowing in, depolarization activates voltage-gated potassium channels, potassium flows out, repolarization, over and over again. This is a story that you should be able to tell yourself and be able to tell me. Lots of new material though, using the principles that you already understand, but with some new detail. Questions? So you might say, well, let me ask myself a question. So calcium is entering through calcium channels. Does the AV node contract? And the answer is yeah, it contracts. It's a muscle cell. This calcium is flowing in. It contracts. But the contraction, the AV node is this tiny little thing. It does contraction of the AV node. That's not its job. Its job is to pace, not to contract. It does contract, but that's not what it's there for. What it's there for is to pace and to cause an action potential that will spread throughout the atrial muscle, whose job it is to contract. So the SA node is the leader, and the AV and the atria are the followers. So the SA node fires the action potential that I've been describing here, and then that action potential spreads throughout the atria in an action potential that I'm going to describe here. Atrial muscle has a very negative resting membrane potential. Left to itself, it would sit at that resting membrane potential for hours. It would never beat. But you know it beats. Why does it beat? Because it gets that stimulus from the action potential spreading out of the AV node, the SA node. So it gets a stimulus, which we can't even see here. Voltage-gated sodium channels, just like the ones in nerve. So now we see the upstroke in atrial muscle is due to voltage-gated sodium channels, just like the ones you studied in nerve. Okay, so the upstroke is the same phenomenon as the upstroke in nerve. Now it gets a little different. Well, no, now it gets a little different. The sodium channels, just like in nerve, undergo the conformational change from activated or open to inactivated. So sodium, after they activate, sodium channels inactivate, and they go into a non-conducting state. The activation of the voltage-gated sodium channels, the depolarization, opens the voltage-gated calcium channels. Calcium flows in, calcium-induced calcium release from the SR, contraction. So this calcium entry is what triggers the contraction of atrial muscle, which is something that atrial muscle does. Okay, so, and calcium channels are slow buggers. They're slow to activate, and they stay open for a long time, which is why this action potential stays depolarized for, a fifth of a second, 200 milliseconds. This action potential is much longer in duration than a nerve action potential. You may remember from self-is that a nerve action potential is maybe 1 millisecond long. This one is 200 milliseconds long because it involves the slowly activating and slowly deactivating calcium channels. So calcium is entering here and the membrane is staying depolarized for 200 milliseconds. So that, and calcium is continuing to flow in, calcium is continuing to be released from the SR, calcium is continuing to be binding to troponin, triggering contraction, atrial systole. Eventually, the calcium channels inactivate, and eventually the potassium channels, the voltage-gated potassium channels activate. They're both slow, and eventually the membrane potential repolarizes back to the resting membrane potential. And it waits for the next action potential to spread from the SA node. You'll just sit there and wait another 800 milliseconds or so, 0.8 of a second. And then it gets the next action, then it gets the next signal from the SA node to beat. And this happens, again, over about once a second. So let's do a clicker. Let's activate the room a little bit. And then we'll take a break. Okay, so this clicker jumps the gun just a little bit because it talks about the action potential in atrial muscle, which I just walked you through, and the action potential in ventricular muscle cells that I haven't walked you through yet. But let me just tell you that the action potential in ventricle is going to be exactly the same as the action potential in atrial. They are exactly the same. Well, they're the same. Okay, so show me your answer to this clicker. During the upstroke of the action potential in atrial and ventricular muscle cells, what ions are flowing and in what direction? During the upstroke, what ions are flowing? The best answer is A. You could make arguments for the rest, but A is the only one that is really right. Okay, so let's do another one. Because I'm moving on because most of you got this right. If you didn't get it right, I'm happy to answer questions. When action potentials spread from one cell to the next, okay, so remember, when that wave of action potential is spreading over the atria, or when that wave of action potential is spreading throughout the ventricles, it's spreading from one cell to the next. So when action potentials spread from one cell to the next cell, what ion flows are important? And I'll give you a hint. There's more than one correct answer to this question. So I see a lot of partial correct answers. The best answer is A, B, and C. Let's walk through this one. Because I saw so many of you chose A. Gap junctions are connecting the cytoplasm of one cell to the cytoplasm of another cell. In order for electricity to flow from one cell to the other cell, it has to flow through gap junctions. The ion that has the highest concentration in the cytoplasm of both cells is potassium. So when one cell is depolarized and that ion is flowing from one cell to the next, the ion is potassium. So that's what A is getting at. But sodium is present in the cytoplasm, so sodium also flows from one cell to the next. That's a true statement. A is bigger than B, but B is a true statement. Is that clear? We have potassium in the cytoplasm, but we also have sodium in the cytoplasm, and so both ions are flowing through gap junctions. Sodium is flowing from outside to inside. Why is that? They're both firing action potentials. So they're both firing an action potential, so sodium must be flowing through open voltage-gated sodium channels. This doesn't happen. So I could throw a curveball at you in sort of a quiz or an exam setting, and instead of talking about sodium flowing from extracellular to intracellular, what if I put calcium here? Would that still be a true statement? Calcium is flowing from outside to inside during the spread of action potentials. That's a pretty true statement because, let's go back. When an action potential when this signal is spreading from one cell to the next cell, so here we were focusing on sodium going in both cells, and here we would talk about calcium going in to both cells through voltage-gated calcium channels. So I could do that. Not so much getting at a quiz, but is the concept clear? Ions are flowing between the cells, sodium and potassium, and ions are flowing into the cells from the outside, sodium and calcium. Okay, let's take a break, and we'll turn this action potential, this electrophysiology, into pumping, into contraction. Action potentialology, we understand we understand pacemaking, action potentialology, and we understand atrial, and therefore ventricular muscle, action potentialology. Now, just to make sure we're all reading from the same page, remember that it is the action potential, well, specifically it's the calcium influx during the prolonged depolarization, also known as the plateau of the action potential. The calcium influx through voltage-gated calcium channels that are causing this prolonged depolarization. Calcium influx, calcium-induced calcium release, calcium contraction. So the red graph here is showing contraction. It calls it tension, but really in the part we're calling it pressure, right? So contraction of the muscle cells getting smaller is what generates this wave of pressure in the atria or in the ventricles, depending on whether we're talking about atrial systole or ventricular systole. Let's not worry about refractory period. Let's just focus on the fact that the action potential, the calcium influx during the plateau of the action potential, is what triggers contraction and the generation of pressure, which is delayed a little bit behind. So electrical events are relatively fast, and pressure events are delayed a little bit. This is one heartbeat. So let's do another clicker. during the plateau, so where are we? We're in, sure, move this the wrong way. During the plateau, we are here. During the plateau, calcium flows. How does that calcium trigger contraction of the myocyte? So this is, okay, so there's one best answer. You have to read all the answers carefully. Give me the one best answer. Calcium flows in through voltage-gated calcium channels. How does that trigger contraction? One best answer. The best answer is D. Calcium-induced calcium release, and then calcium ions bind to troponin, causing the conformational changing, causing the cross-bridge cycling, causing contraction. Okay? Is that clear? So now, let's walk through the sequence of events in a heartbeat. We have talked in detail about some of these. So we're going to start at diastole. The heart is relaxed, it's resting, it is refilling with blood. Then there is the ultra-slow or pacemaker depolarization in the SA node. Mechanism: voltage-gated potassium channels close, IH channels, which are sodium permeable, open, sodium influx, and a few voltage-gated calcium channels open. All three of those are what cause the pacemaker depolarization of the sinoatrial node. Then we get an action potential in the SA node due to the activation of a lot of voltage-gated calcium channels. Remember, the upstroke in SA node was due to calcium channels. Then we have the spread of action potentials in the atria. Through what channels is that spread happening? Gap junctions. Now we have action potentials and atrial systole. Action potential, so now you, in your mind, I'm hoping you're walking through the story of, basically I'm asking you to, in your mind, walk through this story. Action potentials in atrial muscle, voltage-gated sodium channels, voltage-gated calcium channels, voltage-gated potassium channels, and the other channels inactivated. Back to rest, that's the electrical event. Voltage-gated calcium channels triggering contraction of atrial muscle, atrial systole. So that all of that information is embedded here. Now we have this delay at the AV node. The AV node enforces this pause, this gap, time gap, between atrial systole and ventricular systole. The action potentials in the AV node look just like the action potentials in the SA node, with one major exception. Their their pacemaker depolarization is super slow. Why is that good for us? Do we want to have two pacemakers? No, we only want to have one pacemaker. So the SA node reaches threshold and fires the action potential and is the driver of the operation, is the driver of the heartbeat. And that spreads throughout the atria, and then it spreads to the AV node, and the AV node is normally the follower because it didn't reach threshold by itself. because its slow depolarization is too slow. But what's the advantage here is that it will, if it doesn't get a signal from the SA node, it'll reach threshold and fire an action potential. And that gives us a backup. The AV node can act as a backup pacemaker in the case of when the SA node is diseased or damaged. It might be too slow, might not be a great pacemaker, but slow heartbeat is a lot better than no heartbeat. So the AV node has action potentials that look like the SA node, and because they use slow calcium channels, the spread of the action potential through the AV node is slow. Kendra?
So the SA node is its action potential faster than the AV node, or are they about the same?
This part here, is that what you're asking about, this part here?
No, like the overall, like, process.
This part here is about the same. This part here is much faster in the SA node than in the AV node. Yeah.
When you say a slow calcium channel, do you mean like literally slow or just fewer channels?
I mean, it's really kind of both. There are fewer channels, and their intrinsic kinetics of activation is much slower than the intrinsic kinetics of voltage-gated sodium or potassium channels. So the channels are slow, and there aren't as many of them. So now we understand, let's go back to the checklist. We understand the slow conduction through the AV node. It fires slowly propagating, slowly conducting action potentials in the AV node that look like the action potentials in the SA node, but are not usually spontaneous because the SA node is the driver. And the AV node, like the rest of the heart, is a follower. So now we have fast conduction through bundles and then ventricular muscle. What kind of action potential are we talking about? This kind. I described this in the setting of the atria, but it looks the same in the bundles and in the ventricular muscle. Fast sodium channels, fast depolarization, fast spread, lots of calcium, enough calcium to trigger contraction. Contraction of the ventricular muscle, ventricular system. And then there's going to be relaxation. You gotta have relaxation. Where do we see relaxation here? What triggers relaxation? Repolarization. So depolarization and calcium influx triggers contraction. Repolarization and calcium pumping by the calcium ATPase, CERCA and PMCA and NCX, the exchanger, is what triggers relaxation. Clear? Okay. Repolarization, relaxation. We're now back at diastole. Do it again, and again and again, 60 beats per minute. So now let's sort of circle back using our knowledge and this cartoon to pull the anatomy and the physiology together. So you already have this cartoon, and I think what I have now is a series of sort of repeats. So I don't want to, the PowerPoint slide has a series of repeats. It tells the story again. But I get this feeling that you guys have this story. You don't need to repeat it. So let me just jump straight to some quickers, I think. Maybe I don't. Trying to decide what to do here. I think the answer is I want to elaborate just a little bit more on this story. So this is the story. So instead of doing clickers, let me do this in sort of the so-called Socratic method. I'm going to ask a question, and I'm going to ask you to volunteer to answer it. Rather than being clicker, let's just, some brave soul venture a potential answer. Why is the SA node normally the pacemaker? What is special about the SA node that makes it the normal pacemaker of the heart?
It has its own intrinsic rhythm because it doesn't return to a resting membrane potential. It sort of continues to cycle around the depolarization and repolarization.
Yeah,
And the AV node is a lot slower.
right, okay. So the SA node has an intrinsic depolarization mechanism. Potassium channels close, IH channels open, a few calcium channels open. It spontaneously depolarizes to its threshold and then fires a slow action potential, which then spreads. So that's what makes it the pacemaker. None of the other cells in the heart normally are pacemakers. The atrial muscle has a negative resting membrane potential. The AV node, which also has a slow depolarization component, is too slow, normally. It's a backup pacemaker. Okay, so I'll ask another question. When SA node fires an action potential, are voltage-gated sodium channels involved in that action potential at the SA node? Do we need voltage-gated sodium channels for the action potential in the SA node? No, they don't play a role. When action potentials, what do we need? We need voltage-gated calcium channels. When action potentials spread throughout the atria, and we trigger atrial systole, I'll ask a different kind of question. Where is the blood pumped? When the atria contract and they generate pressure, where does that blood flow under the pressure being generated by the atrial systole? Kendra? Into the ventricles. The blood in the right atrium is pumped into the right ventricle. The blood in the left atrium is pumped into the left ventricle through the open tricuspid and bicuspid valves. Michael?
Are there valves preventing backflow for that one, or?
There is not. So, in fact, contraction of the right atrium causes some regurgitation of blood into the inferior and superior vena cava. And contraction of the left atrium causes some regurgitation into the pulmonary veins. We don't have, oh, there is some regurgitation. It's not big, but it's some. Okay. Why doesn't this electrical activity spread right here from the left atrial wall into the left ventricular wall. Great.
There are fibrous rings between the atria and the ventricles that act as insulators.
Insulation. There is an abnormality where if that insulation is incomplete, there can be spread. This is a disease. This is a physiology class. We're not going to talk about pathophysiology too much, but there is a syndrome called Wolff-Parkinson-White syndrome where action potentials can spread from here to here, and then the heart beats abnormally. It's an abnormal heartbeat. And usually that's treated with a surgical approach to try to break the connection here, break the electrical connection. Why does the AV node delay the action potential spreading from atria to ventricles? Kendra?
It has a slower, like, action potential.
It has a slower action potential, and action potentials move from cell to cell to cell slower. What is the order in which the different parts of the ventricle contract during ventricular systole?
The septum to the apex and then to the walls of the ventricle.
Exactly. Septum, apex, walls. And we'll need that for understanding the ECG, the electrocardiogram, on Thursday. Kendra?
I have a question. Is it just, so at the apex, those are also Purkinje fibers?
So, yeah, okay, here's a question. What's the difference between the Purkinje fibers that are sort of, it's not even shown very well in this image, but at the end, well, these things here, these little fibers here, what is the difference between a Purkinje fiber and a ventricular muscle cell? I didn't teach this. The answer is not much. It's just that there is this sort of conduction pathway, branches and Purkinje fibers that help move the electricity throughout the big, thick ventricles in a well-organized pattern. So that, and because of that pathway, we get septum, apex, free wall contraction in that order. Why does that help us with the heartbeat? Well, it kind of does. So when the septum contracts, you'll see this, I think, in the frog heart, even though it doesn't have a septum. So I don't understand. Well, so the septum, when the septum contracts, it both contracts this way and it contracts this way. If you've ever seen a movie of an actual human heart beating, it moves this way first and then moves this way and then contracts. So it moves up and then in as during ventricular systole. And that moving up is due to shrinking, shortening of the intraventricular septum. Okay, what channels are most important for triggering contraction in atria or ventricles? Joe?
The voltage-gated calcium channels and the potassium channel.
Voltage-gated calcium channels.
And, that was what Joey said. It's not a voltage-gated channel. It's calcium release channels. Calcium-induced calcium release is the second most important channel for triggering the contraction. of both muscle cells. Did I ask the question in a way that led you to not think about calcium-induced calcium release? I mean, it's there, it's a channel. It doesn't happen to be in the surface membrane, but it's a channel. What is the channel that is most important for causing the transition between ventricular systole and ventricular diastole? What's the most important channel for causing the relaxation of the heart after systole?
Potassium channels.
Voltage-gated potassium channels. They trigger the relaxation, the repolarization, which closes the voltage-gated calcium channels, which causes the less calcium to flow in, so the calcium release channels close. What causes calcium to get pumped out of the heart, which is what is necessary for transit out of the heart cells, which is what is necessary to cause the transition from contraction to relaxation. What pumps pump calcium out? Circa, PMCA, and NCX. Like,
I remember a long time ago, you said something about NCH looking backwards.
yeah, okay, I thought that was gonna come up. It wasn't so long ago, it was like last week, right? Can't have been too long ago, I mean, we're only in the third week of the semester. Is that even true? Is this the third week? Okay. NCX pumps one calcium ion. So I want to answer the question because it's interesting, but I don't want to get it caught up into too much detail. NCX pumps one calcium ion out in exchange for 3 sodium ions in. It's a 3-to-1 ratio. We need 3 sodiums moving in order to provide enough energy to pump 1 calcium out. So we have 3 positive charges moving in for every 2 positive charges moving out, because calcium is divalent. So the NCX is what's called electrogenic. It senses the electric field because it's every time NCX moves, a charge is moving inward. Every time it pumps calcium out, a charge is moving inward. Bottom line, when the membrane potential is polarized, NCX allows sodium to flow down its concentration gradient and pumps calcium out. When the membrane potential is depolarized, it runs in reverse. It pumps sodium out in exchange for letting calcium in. That is not a major player in the calcium influx here, but it does happen. So NCX is pumping calcium out here and is actually pumping calcium in here because of its voltage dependence. That is a upper level thing. I will not test you on that detail of NCX. But that's the answer to that question that has been keeping you awake at night for a week now. Okay, so that's the heartbeat. We've really walked through it, both from an anatomical and from a physiological perspective, right down to the cellular and molecular details, okay? But I know it takes some time to settle in. So you need to work with it, think about it, write the story out, make sure you can explain to yourself what is a pacemaker cell, why do ventricular muscles and atrial muscle cells contract, why does the action potential spread fast in ventricular muscle and slow in the AV mode. Those are the kinds of details. The book will help. Remember, the book calls it IF, not IH. It's the same thing. So just last point, IH is an informative name because the H stands for hyperpolarization. IF was a name that was given to it by by electrophysiologists when they discovered it. They discovered a channel that was opened by hyperpolarization. And they said, huh, that's funny. hence the name F. It was the funny channel, but then it became less funny. It became the hyperpolarization activated channel. The F stands for funny. Okay, enough for today. On Thursday, we will, problem sets. So I'll give you your problem sets. I want to talk about them for a minute, so let me just say a few words. I'm only going to say a few words. One is that the place where most of you lost the most points was in the circling or underlining or highlighting of the proteins in the story. So the beta-gamma complex is a protein. The alpha subunit is a protein. Substrates for kinases are proteins. Nobody got that right, but I took off a point for not, no, that's not true. There's one person who got that right. Regulatory subunits are proteins. Catalytic subunits are proteins. Cyclic AMP is not a protein. So just go through, you'll see, the answer key is posted on Moodle. Go through and see where you lost your points. Most of you did pretty well. B plus, A minus sort of thing. So but use this to help, see what you get and what you need to work on something. Let me hand these out.